Recording. Very good. Let's hope some good internet. Let's open it up. All right, so let's start with quiz 1 review. So I'm doing these quizzes so that you stay sharp on your studying. This should help you gauge how well you're doing as you prepare for the exam. I made this quiz very approachable. You should have been able to only study the PowerPoint and do relatively well, like a 6 or above on this quiz. I'd say if you just studied the PowerPoint and attended lecture. Read the book, maybe I'd expect like an 8 or 9. Nobody got a 10. Somebody did get a 0, but nobody got a 10. If you got below a 5 or a 6, I'm going to say ramp it up and talk to me if you need help studying. But if you got a 5 on that quiz, something's going on, so we need to address that. Because we're going to take a quiz or an exam every week, again, just to keep you sharp. Can't be falling behind. So let's take a look at this, and I can see about how well, or let me think about how to rephrase this. I can tell that about 75% of you are staying on top of it and doing well, paying attention during class, maybe cracking your book open every now and then, based on the responses here. So, who proposed germ theory? I told you so many times that I would ask this question. 3/4 of you listen. So, the other quarter of you, get it together. When I tell you I'm going to ask you something, I'm going to ask it. All right? I'm going to do that a couple times today. When I do it, I'm going to straight up tell you, write that down. This slide will be on your quiz. This will be on your exam. This is proof. It's going to keep coming up. So same thing with this. I told you that I would ask about this woman. This is the only woman that we spoke of during lecture 1, Franny Hess. So she's the one who was working with Coke, who suggested we don't use gelatin. We use agar because it's more durable under heat. Gelatin will melt at 37 degrees. Agar will not. 37 degrees is human temperature, so we're going to grow bacteria at the temperature that they would grow in humans to see how we could reproduce and infect. So again, about 3/4 of you got this right. I told you I'd ask it. 3/4 of you listened. This is based on a table that I told you I would ask about. So if you gain or lose a neutron, you're an isotope. If you gain or lose what will we call you an ion? An ion gains or loses what? Electron, perfect. What is a molecule? To what? There you go. So 2 or more atoms put together, and they can be the same or different elements. So for example, O2 is molecular oxygen. H2O is a molecule of water. And then what is going to differentiate our elements? Different number of what? Protons. Right. And keep in mind, protons and neutrons are in the nucleus. Electrons are outside in what we're going to call electron fields or shells. Okay, again, straightforward definition. Ionic bonds are formed when one atom donates an electron to another. That's going to create an electrical attraction. So one becomes positive, one becomes negative. Therefore, they will join through electrostatic particles. Somebody explain a covalent bond. Sharing what?
Share their electrons.
Right. In covalent bonds, we're going to be sharing electrons. Remember that electrons that are available in the outer shells, we call those valence electrons. Covalent means that we have togetherness within our valence. You are sharing your outermost electrons. Covalent. And hydrogen bonds are only going to take place in hydrogen. Glad nobody answered that. All right, told you I'd ask you this one. Primary sequence is just a chain of amino acids bound together by what type of bond?
Peptide.
Peptide bond, right. I do want you to know that. And keep in mind that the peptide bond is created through the dehydration synthesis, also known as the condensation reaction. And I believe I added that slide to the beginning of what was it, day 4, day 3 or day 4? This is the follow-up day. So that was the first slide on there. All right, this was, I'd say, the hardest question on here. So Schleiden and Schwann were the ones who we credit saying that the cell is the fundamental unit of life. So this is our definition that we're going to go back to and say a virus is not alive. We can go all the way back to here. So then Virchow came up about 20 years or so later from Schleiden and Schwann and said, all right, if that's the fundamental unit, the cell, all cells are arising from other cells. So 2 of you got that. The most inclusive is going to be a domain. What is the most exclusive? A species. Perfect. All right, so cocci, these can form the most abundant type, the most diverse type of cellular arrangement. Keep in mind that cocci is a shape. What is the word that we're going to use to describe bacterial shapes? Starts with an M. Morphology. So with bacterial cells, we have morphology and arrangement. Morphology is coccus, bacillus, spirilla. So these are shapes. And then do they exist in chains, as individual cells, clusters? That's going to be the arrangement of the shape. So just following up, if you have this question, you could be able to make another set of questions. So what is the, of these, which has the least diverse cellular arrangement? Somebody take a stab at that. So I would say spirilla or vibrio, because they both only exist as a single cell. There you have 2 options. Bacilli can exist in 3 arrangements: single cell, chains, or as the 2-cell organization called diplo. That'd be diplobacillus. Okay, I was kind of surprised by the responses to this, so I think this is really important that we talk about. So I understand this was on Thursday, the day we took the quiz, but this shows me that a handful of you aren't reading before you come to class. Again, if you're going to read after class, I definitely recommend that you read, at least open the book before class, especially on Thursday, since we're going to have quizzes on Thursdays. So, all cells are going to have ribosomes. That cannot be correct. What does a ribosome do? It's going to make proteins. You can't survive without proteins. So, a ribosome is going to synthesize proteins from the DNA, ultimately. Gotta have it. All cells, eukaryotes, prokaryotes, all of them have a plasma membrane. And if we're talking about bacteria, all bacteria, to my knowledge, are going to have peptidoglycan. Some have more, some have less, but we're going to have peptidoglycan in the cell wall. So the difference is that LPS, this is only in gram-negative. No gram-positives are going to have this. All right, and then this one was pretty straightforward. Pretty happy with this. So the genome is a complete set of genes in an organism. If we're talking about bacteria, why is it not necessarily appropriate to say that the chromosome is the genome? What else would we have? Huh? There you go. Right, we can have a plasmid. So keep in mind we have pili. Those conjugation pili can exchange genetic material from one bacterial cell to another. You could end up with, say, a plasmid for antibiotic resistance that's not in your genome, but it's within your cell, so it's a part, not in your chromosome, but it is a part of your genome because it's within your cell. Also, keep in mind that humans have more than one chromosome. We have linear chromosomes, 23 pairs of them, so 46 total. So our genome is more than one chromosome. All right. We're going to do the Respondus lockdown browser from here on out for all quizzes and exams. So I have links here. I don't have these posted in the slides available for you all, but I did send out an email with these links. So you should be able to go to this link here at the bottom to download this. If for whatever reason you have trouble with this, go into quizzes in Canvas in our page, and then there's one that says test quiz Respondus lockdown browser. Click on that, and then it'll take you to this link right here. So we're going to do this for Thursday. So between now and then, try and get it. Let me know if we have issues. All right. So, feeling good? Question? Boom. There we go. All right, you all. So, sorry I hadn't been sharing my screen. What we had done is we just talked about all of the quiz questions. And again, these are available for you. These are the links to download the Respondus lockdown browser. I sent these in an email. So people online, do you all have questions before we get started on chapter 5? All right, take that as a no. How about y'all? Anything? All right, let's do it. Get my phone, take a look at time. All right, we're right at 10. I think we'll probably take 40 minutes or so to get through this. All right, so the chapter starts talking about the cell cycle. We have 3 phases that you all should be concerned with for bacteria. So in the B phase, this is the beginning, the cell is going to get larger in size, and it's going to get ready to replicate its DNA, its chromosome. At that point, we're going to move into this C phase. Think C chromosome. We're going to replicate our chromosome during this phase. And we're going to end up with 2 identical chromosomes within one cytoplasm. After that, we're going to enter the D phase, where we're going to divide. So C chromosome, D divide. And maybe B, you can think before. So during the D phase, what we're going to end up with is this fission ring apparatus. So it's going to enclose in the middle of the cell and create a new cell wall so that we have 2 daughter cells that are genetically identical to one another. And then after that takes place, we're going to go through a process called binary fission. So fission means to separate, binary meaning 2. We create 2 identical cells from one parental cell. So when we say that bacteria and archaeal cells can grow exponentially, what we're referring to here is when we undergo binary fission, we go from 1 cell to 2 cells, 2 cells double and turn to 4 cells, 4 turns to 8, 8 to 16, and so on. So every time that you double, you end up with more and more cells. And if you were to look at a growth curve indefinitely, it would look something like this. This isn't what you're going to see in a lab setting, but should you just keep giving them nutrients and getting waste products out, they would just continue to grow. So every time that a cell divides, we're going to call it the generation time or the doubling time, because again, every division we end up with twice the amount. So we call it the doubling time. So if we're talking about pathogens, if an organism divides really rapidly, really quickly, you're going to end up displaying some symptoms of an illness faster than something that grows more slowly. And the incubation period is the time from which you acquire an infectious agent to the time that you display symptoms of a disease. We call that the incubation period. So how long does it take for that organism to cause a problem in your body? And I'd like for you all to keep in mind that all organisms are different. So something like E. coli, the book says it divides in 15 minutes. I've always heard it be about 30 minutes. But either way, this is really fast. So a quarter to half an hour for E. coli to go through the whole cell cycle. That's really fast. Whereas an organism that I've done a lot of work with, called P. gingivalis, this is an oral pathogen, causes gum disease. This takes 4 hours to grow, to divide. So this is 8 to 16 times slower, and it's anaerobic. So all organisms are different in their requirements and how fast they grow. All right, we have 4 distinct phases of cell growth that we're going to talk about. I will ask you about this. So this is where you put a little thing, test or exam question, we'll ask about this. Okay, so we've talked about broth a handful of times. Broth is liquid growth medium. When I say growth medium, I just mean it's food for small life. So it's bacterial food in a liquid form. Bacterial yeast food. So you take a single colony and you're going to inoculate. That's a word that we're going to use to add life to a sterile environment. We're going to say you inoculate something. So you add bacteria to growth media. We've now entered the lag phase. So this is where, so here on the x-axis we have time, and on the y-axis we have number of bacterial cells. But keep in mind these are viable cells. So viable means cells that are alive, as opposed to dead cells. Viable. So these cells in the lag phase, they've just been exposed to this new, really rich medium most likely, and they're going to be in their B phase getting ready to divide. Okay? So they're going to grow inside, they get ready to divide, and then when they do, we start to see more viable cells, more of them as they divide. And we're going to enter what's called the logarithmic, or shortened to the log phase. This is the exponential growth phase of a bacterial growth curve. So the cells, they're just rapidly dividing, going crazy. And we're going to call the broth, as these cells divide, it's going to get kind of cloudy. The word that we're going to use to describe that is turbid. T-U-R-B-I-D. Turbidity is cloudiness within a bacterial sample or a microorganismal sample. So if you wanted to grow organisms and then put them on a petri dish and grow them on an agar plate, this would be the time when you would want to get them, during the log phase. Because they're dividing really well. You take them and put them on a solid medium, they're going to keep dividing and turn into individual colonies. And I'll show you what that looks like. We've got a couple images here. So then you'll notice that we're going to hit a plateau here at the end of the log phase. And you can't see it really clearly, but at the top of this, this is red. At the bottom of the tube, it's kind of a brown color. And so what this indicates is live versus dead cells. I'm a little bit hesitant on this, honestly, but the concept is applicable, I'd say. So what they're saying is that once you enter the stationary phase, some of the cells are going to die. And this makes sense, right? You've consumed pretty much all the nutrients available and you've stopped growing exponentially. You're also starting to build up some waste products that can inhibit the growth further. So you have waste products, you don't really have nutrients available, you're running out of space. So we end up with some dead cells collecting and some live cells present. I'd say this fraction of dead cells is going to get larger as you're longer in the stationary period. Makes sense, right? But the point being, you're going to have a whole lot of alive cells during the log phase, more dead cells in the stationary phase. After that, you're going to enter the decline or the death phase. So you've really built up your dangerous end products of metabolism. You've run out of nutrients. Your cells are just starving at this point. It'd be like if you stayed home for 4 weeks without any food, you're not going to make it any longer. Same thing here. And point being here, during the log phase, I just want to keep this rolling before I switch slides. This is when bacteria are most susceptible to antibiotics because they're actively dividing. A lot of antibiotics work on replication machinery. Not all of them do, but some of them do. All right, and then somebody last week told me I was going kind of fast, so I can see how you all are doing and you all are taking notes, but people online, if you want me to slow down, just interrupt me and just say something, okay? All right, and then these are the last two phases that we just talked about. I forgot that I had included this slide here, so we've gone through this whole cell cycle. Questions about this? Again, I'm going to ask you about this, so be ready for it for the quiz on Thursday. All right, so that is a growth curve, generally speaking. So let's talk about what happens if bacteria do get exposed to something dangerous like an environmental toxin or an antibiotic. So some cells can go into a state of dormancy or non-dividing. So it's kind of like hibernation for bacteria. So take a look at this figure. Let's say that we expose our bacterial cells to an antibiotic, and we wipe out most of them, let's say 99.9% of them, but one or two cells survive. Those cells that have survived have some sort of resistance to them. We're going to call those persister cells. So those cells recognize that something's going on in the environment that's really dangerous to them. So what they're going to do is they're going to lay really low, turn the metabolism down to almost nothing. Again, think of this as hibernation for bacteria. So they're going to stay in their B phase of the cell cycle, where they're just waiting for the environment to change so that they can replicate and divide, increase their population size. So then whenever the conditions become more favorable, those antibiotic-resistant cells, or whatever those persister cells were, those are going to undergo cell division, binary fission, and repopulate. So again, this is how we can end up with some antibiotic-resistant strains. These persister cells can, through conjugation, share DNA with other cells. And this is how we can end up with things like MRSA, the methicillin-resistant Staph aureus that we see in hospitals. One other way bacteria can deal with non-favorable conditions is by producing endospores. But only certain gram-positive organisms can do this. Somebody online, tell me the difference between gram-positive and gram-negative bacteria. Anybody?
Gram-positive doesn't have any bacteria or exposure to any of that, and gram-negative does.
Okay, so gram-positives don't have, so this is on the quiz, what do gram-negatives have that gram-positives don't have in their cell wall?
Don't they have, the difference is a thick layer and thinner layer?
Right, so gram-positives have a thick peptidoglycan layer in their cell wall, but they lack lipopolysaccharide, or LPS. That is only present in gram-negatives. Thank you for trying, though. I really appreciate your effort. So gram-positives, thick peptidoglycan, gram-negatives, thin, and they have LPS. Gram-positives do not. Just to reemphasize big points. Gram-positive, gram-negative is something you all should leave this class understanding. So only some gram-positive organisms in the genera, or the plural of genus, it is Bacillus and Clostridium. So these can produce endospores. So one way that they can tell whether or not they're going to need to create an endospore is through quorum sensing. So we talked about quorum sensing in relation to cystic fibrosis. So you can sense a lot of your nearby neighbors and say, all right, now I'm going to cause problems in a human. Well, if you send out these signaling molecules and don't receive them, that says there's none of me nearby. So my numbers are low. All right? So it'd be like me just throwing let's just say we all walk in the room and we're throwing golf balls around. If I don't get hit by a golf ball, that means that there's no other people throwing golf balls around. So I can say, all right, something in the environment is bad because I should be receiving signaling molecules, but I'm not. So I'm going to make a spore. So you can see is we have this cortex wrapped by a coat. All right, so this is the process. So let's walk through this. So what we're going to do first is we're going to end up with our DNA replicating, and then we're going to put that towards one end of the cell. And endospores can form on the ends, in the middle, or somewhere in between. They can form in different places within the cytoplasm. So we're going to form our replicated DNA. We're going to form the septum here and then wrap it in a cortex. The book mentions this, pardon my pronunciation here, dipycolinic acid. So this is in the cytoplasm of the spore. This is going to help maintain the integrity of the DNA and the protein. So you have DNA, you want to keep it protected. We have a certain acid within the cytoplasm to do that.
Hey, Nick, where are you on this cycle, on this picture?
Yeah, okay, so right here, you can see can you see my cursor?
No, we can't see anything, or I can't see anything.
Okay. Gotcha. So let's see, I'm using my cursor, so if we start at the top where it says vegetative cell, let's start there. So vegetative cell, by definition, that means an actively dividing cell. Think vegetation, green, life, vegetative, as opposed to dead. So a viable cell can be dormant or vegetative. Dormant, hibernating, vegetative, actively growing, dividing. Both of those mean it's not dead. So moving from the vegetative cell at the top here, we go to the right where it says transverse septum. So we're going to pinch off the DNA there, and then we're going to wrap it with a cortex. The cortex is peptidoglycan. So we're forming a cell wall around our newly formed chromosome. Then we end up down here at the bottom of this figure 5.8. It says spore coat segments. The spore coat contains about 70 proteins. That's going to be outside of the peptidoglycan. Then we're going to end up in part C on the left here, where the mother cell that created the spore is going to rupture because it senses something in the environment, so it's going to slow down its metabolism. It made a spore to do its job. Its job being to keep its DNA going in the next generation. And then at that point, we end up with a free spore. That spore is resistant to just about anything you throw at it. So desiccation, meaning it can resist being very dry. So I don't know if these can go to space and survive coming back. They probably can. Also heat, it's very resistant to heat. And this whole process takes about 6 or 8 hours, which seems really fast, but if you think about the doubling time being maybe 30 minutes to an hour, this takes a long time, relatively. So the book says this is not a reproductive process. I'm going to argue against that. Why is this not a reproductive process? We are creating new DNA from parental DNA so that we can become a new vegetative cell. So I would argue the book's wrong on that. This is a reproductive process. We're making new DNA. So that's why I have it in quotes. The book says it's not. I'm going to argue against that. This is reproduction. It's just a different kind of reproduction. So we have a spore, and that spore is there so that when the environment gets better, less damaging, less harmful, it can come back to life and become a vegetative cell again. And that's going to take about an hour and a half for those proteins to break down in the coat and our new cell to start dividing again. So how resistant are these? You can boil them in water for 2 hours, right? You know, whenever we have the water advisory warnings and we boil our water for maybe 10, 20 minutes, spores are still going to survive that. Let's see. They can survive 70% ethanol, so what we use in every clinical setting to clean stuff with, 20 years. That's outrageous to me. If they can survive 20 years, they can probably go another 10. Why not, right? But this means that somebody's done this. Somebody's put spores in a jar with ethanol for 20 years and said, yep, it's still alive. So that's a long experiment. They can survive 1 million rems of radiation. I'm not a radiologist, but humans can only survive 500. So these spores, very resistant to UV radiation. And so how do we get rid of them? The only way we can do this is to autoclave. So an autoclave is a box that gets, we're going to take water and create steam to create really high temperature, high pressure environment for a long time. So steam, high pressure, high temp, long time will kill spores. It's the only way we can do it. All right, this is also a candidate for Thursday's quiz. So here we see the 3 domains of life. We have archaea, bacteria, and eukarya. And we can see different ranges of temperatures that these organisms can withstand. Eukarya being the less diverse, and archaea being the most diverse as far as temperatures that we can survive at. Shouldn't come as a surprise. But I think this is really important, so I would like for you to know this. And then the next slide or two, we just have these curves laid out in some definitions with some numbers as far as the temperature ranges. So again, if we're just going through and just having a series of definitions, I want you to study these definitions, so this is a good candidate for Thursday's quiz. So this is figure 5.9. All right, so going through definitions. Psychrophiles, these are going to be organisms that grow below 15 Celsius. So these are things that like really cold environments. So if these like 15 degrees Celsius, they're not going to live inside us because we're about 37 degrees Celsius. So these are not going to be human pathogens. Again, the point of this class is to understand what makes us sick mostly. So psychrophiles, not going to be it. We need to worry about mesophiles. These are organisms that like, I don't know, 10 to 45 degrees Celsius. Again, we're 37 degrees Celsius. Mesophiles are going to be human pathogens. Thermophiles are going to live in really hot areas, so like 60 or above, so hot springs, compost piles. And these can survive pasteurization. So pasteurization refers to Louis Pasteur, right? Pasteur is the low and slow heating of food products. So milk has been pasteurized unless it's raw. Juices are usually pasteurized. So we're not boiling it, but we're going to put it up to about 63 degrees for a long period of time. And that's going to kill most of the stuff that's going to make us sick. Certain mycobacteria will survive that, though. But you're probably better consuming something pasteurized than raw if you don't know where it's coming from. And then archaea, we can classify as hyperthermophiles if they're living on, say, like the sea floor at near boiling, right? Water boils at 100 Celsius. These are living at 80 Celsius. Okay, so that's temperature. Let's go into oxygen. So there's a cool test called the aerotolerance test. We're going to use a medium called thioglycolate. I expect you all to do this in your lab. Today's really applicable to your lab stuff. You are going to do most of these tests probably. So paying attention here is going to help you with your lab. So if you require oxygen, we're going to call you an obligate aerobe. You must have, you are obligated to have oxygen. Obligate aerobe. So let's talk about this medium real quick. So this thioglycolate, you'll notice it has two colors here. So it has a dye that is an indicator for oxygen. If it detects a lot of oxygen, it's going to be purple. Less oxygen, it's going to be yellow. So you open the tube, and then, we have a lot of oxygen in our environment, so this headspace has O2. It turns the top of the medium purple. Oxygen doesn't go really far down in this tube unless you mix it up, right? So it's really important to not mix this medium. You're going to end up with yellow as an anaerobic or less aerobic region. So based on the way that organisms grow in this medium, we can detect their oxygen requirements. If you have to have oxygen, you're not going to be growing down here because that's anaerobic. You would die. So something that requires oxygen is only going to be in this purple layer at the top. Microaerophiles are going to live in low oxygen environments. So where do you expect to see them out of these 5 figures or 5 subparts here? Where do you expect to see a microaerophile? Yeah, I'd say B.
B.
There you go, perfect, thank you. So right at the interface of the high and low is where we'd expect to find a microaerophile, where we start to have less oxygen.
So is this where hyperbaric chambers come in? That you want your,
I'm sorry,
the hyperbaric chambers, you want your cells to grow rapidly, so they put people in those?
I'm sorry, what is a hyperbaric? Are you referring to like a high pressure?
Yes, the oxygen I don't know. It's like a big oxygen tanning bed. Have you never seen that?
No, I have not seen that before.
Okay, never mind.
Okay, gotcha. But just to address your question the way that I can, this test has nothing to do with pressure. These tubes are exposed to just the atmosphere that would be in a lab. No pressure is involved here. And we can only tell about oxygen requirements. That is all that this test gives us. So C are going to be your obligate aerobes. B, microaerophiles. Your anaerobes are going to be, I'd say somewhere here in like E. If you are an obligate anaerobe, you have to avoid oxygen. You're not going to be in the purple layer. Whereas some organisms, as we'll see on this next slide, are called facultative anaerobes. Those are going to grow throughout the whole media. So that means I like oxygen, but I can survive in low oxygen too. So I can go anaerobic, but I prefer oxygen. That's a facultative anaerobe. And we're going to find those throughout the whole media. So aerotolerant species, insensitive to oxygen. So again, that means they're not going to use oxygen, but it doesn't really bother them too much. Whereas an obligate anaerobe is not going to survive in the face of oxygen. And then these capnophilic bacteria, they're going to require low oxygen and high carbon dioxide. So the book has this image with a candle in it. I have never, ever seen this done, practically speaking, so I don't know if this is outdated or if I just haven't studied these bacteria. So I assume what's going on is the oxygen is consumed by the flame, right? So fire requires oxygen. So then the flame will go out when the oxygen is low, and then I assume that the soot that comes off of the wick creates more carbon in the environment. So I assume that's what's going on with this test. Otherwise, we're going to use these hydrogen gas packs that you can see here in this second part B. So these gas packs are going to eliminate oxygen from the environment. They're going to increase the concentration of hydrogen gas. And then we put these in some airtight jars and seal them off. And you have to replace this pack every day, every 2 days. Or you could use this really cool anaerobic incubation chamber. So this is what I worked with in the past. you end up putting your hands in these large gloves because this whole box is anaerobic. So there's hardly any oxygen in this entire box here. And so this is kind of like working in a space station. You have this as an airlock, so you put your samples in here, close this door, exchange all the gases, and then open another door on the inside. So this is a way to suck out all the oxygen before you put stuff in. So this way you can manipulate a large amount of bacteria at once without using gas packs. Here you would need to use tanks of gases. All right, going through just more definitions. We've talked about these already. Acidophiles like acidic pH. Somebody tell me acidic pH range. 0 to 7, right. If you like basic conditions, you are called an alkylophile. Remember, alkaline is the same as basic. So your acidophiles, these are things like lactobacilli that we're using for our dairy products and such. Certain organisms can withstand high amount of pressure, so barophiles, they love pressure. So the psychrophiles that are living at the bottom of the ocean in those really high temp areas, so like 80 Celsius, there's a lot of pressure down there too. Talked about halophiles, love salt, live in the Dead Sea, you're a halophile. And then this last slide here, so if you're a halophile, you can withstand a lot of osmotic pressure. You have a lot of solutes on the outside of your cell compared to the inside of your cell. That's what that means. So which way would solutes want to go? What? Yeah, so solutes would come in, water would want to go out. So you would end up desiccating. So they're resistant to that. So they can maintain their water within their cytoplasm, even though there's a lot of salt, a lot of solute outside of their cells. All right, so let's talk about some culturing techniques. Generally speaking, we have two types. We have, as far as media is concerned, we have undefined and defined. So undefined medium contains something like beef extract, yeast extract. It's just, we have proteins, right? But we're not going to say it's this protein and there's this many milligrams and this many liters, right? It's just, we took cow and turned it into a powder so that you can give your bacteria some protein, okay? We do this from time to time. So we'd say that's undefined as opposed to defined, which is you have this protein in this amount, you have this sugar in this amount. It is an exact recipe for you. You all will do this in your lab. So we have selective media versus differential media. You'll probably spend whole labs about each of these, honestly. So a selective medium, this is going to select or think certain things are going to die, other things are not going to die. That's what think natural selection, artificial selection. Can you survive high salt? Can you survive an antibiotic? If you can, you'll be selected for it. If you can't, you'll be selected again. So we're going to see what can you deal with in your environment in selective medium. In differential medium, we can look at biochemical processes. So we can put a certain sugar in there and say, can you use that sugar or not? That does it kill you or not? We're not answering that question, but can you use that or not? Like lactose, for example. And then we can add certain, again, a dye and indicator. So if you can ferment lactose, you're going to end up making an acidic byproduct. So that means your pH is low. An indicator can change color with pH. So let's say that you can ferment some lactose, you might turn out pink, but if you're really, really crazy at fermenting lactose, like E. coli, you'll end up as like a shiny green, like metallic colors. So based on the color, we can say whether or not you can do a biochemical process. There we go. Share. All right, we're rolling. All right, we've talked about pure cultures a little bit. So we have pure cultures as opposed to mixed cultures. A pure culture is going to be only one organism. There's a lot of different cells present, but it's only one species of organism, or one strain, we could say. As opposed to a mixed culture, which is what we're seeing here in this plate. You can see these different colony morphologies. Some are little and yellow, some are larger and white. This shows us that we have two different types of bacteria. This is a mixed culture. A pure culture would have all the same colony morphologies. So one way to do this is the pour plate method. I have never personally done this. The way the book mentions this is you take your liquid medium with the agar, and so it's still in the liquid form. It's like it's still hot, right? So you all know a gel before you put it in the fridge. It's like that. Put your bacteria in when it's not too hot, because you don't want to kill them, and then pour it in a plate. That plate will solidify over a handful of hours overnight, and bacteria will grow on the top. I've never done this personally because I wouldn't want to expose bacteria to a high temperature, so I would avoid this. You all will do this in your lab. This is the streak for isolation, or the streak plate method. The whole idea here is to obtain a single colony. If you want to create a pure culture, you need to do this first. So we're always going to incinerate our loop. A loop is a tool used to move bacteria from one place to another. You expose it to a flame over a Bunsen burner, it's going to get really hot. And then you're going to let it cool, because again, you don't want to kill your bacteria. So get really hot, let it cool, and then you're going to put your loop in your mixed media, in your media with your culture in it. Then you're going to spread it out on a plate, and then from your spread, you're going to go into one of your lines and continue it. The whole point being here is to dilute it every time that you make a new set of squiggles. Then you'll end up with a few colonies in your last, what we're going to call quadrant, your last streak. And then you take this one little cell here with millions of bacterial cells, or one cell, one colony, and put this into a new broth. Now you have a pure culture instead of a mixed culture. This is how we isolate genetically identical organisms from a mixed culture. This is big. I'll probably ask you about this too. This is really important. So I want you to know this going into lab. So how do we isolate a pure culture from a mixed culture? The streak for isolation. All right, let's talk about how we're going to count bacteria. So we can do this directly or indirectly. One way to do this is to apply a stain to bacteria, because again, they're pretty much all water, so you can't really see them in a microscope unless you stain them. So you apply a stain and then put them in this grid. So you would inject like 5-10 microliters here, and then literally count the cells in a grid. Based on the volume that you added and whatever dilution factor you used, you would just multiply back. And if you counted, say, 100 cells, you do your math in reverse, and you could say, in my tube, I had 400 billion bacterial cells. So you're directly counting here, but you're counting cells that are alive and dead. The book also mentions this most probable number test. Again, personally, I've never done this, but I'm going to walk you through what we're going to do. So the samples, we're going to dilute them either 1 to 10 or maybe 1 to 100. Basically, we're going to dilute an original sample of water. We're going to use this to test water quality. So if I drink a certain water, say lake water, pond water, will it make me sick? Is how we're going to, that's why we would do this test. So you dilute your samples to different dilutions. You're going to add it to a broth that contains lactose. Again, E. coli, our gut bacteria really like lactose. So if a pond sample has these enteric bacteria in it, they can make us sick. That's what we're looking for here. So fermentation is going to produce gases. It can change the color of our media depending on the dye present in it. And then based on the reactions that we get, we can say there's more or less bacteria. That's what we're going to say here. We're not counting. We can say there's more or there's less. So you take your sample, and you're going to put different dilutions in your tubes with your media. You'll notice these little rectangles here. This is an inverted glass tube. This is called a Durham tube. You'll see this in your lab. So this is used to detect fermentation. Fermentation is going to result in acidic end products, which are going to change the color of your media. It's also going to produce gases. So things like CO2 are going to result. So fermentation, just like making alcohol, right? CO2 is a byproduct. That gas is going to go from this inverted tube. It's open at the bottom and closed at the top. The gas will displace the liquid. You'll see an air bubble up here. So air bubbles and change in media color indicate you have bacteria that ferment lactose. That tells you don't drink that water. Could be dangerous. You want to treat that water before you consume it. That's why we do this test. All right, we could also do, take a whole plate and spread a sample across it and just count the number of colonies on a plate. You'll notice these are all the same. This is a pure culture. We can count these and then say, all right, well, if I added 1 ml at this dilution, again, I can estimate my numbers in my original sample. And what we're counting are colonies, but when we go back into our math, we're going to call them colony-forming units, because this one colony that you can see here, visible to the eye, contains millions of cells. Those cells originated from one or more, maybe 1, 2, 3 nearby cells. They divided on top of each other and form a colony. So it's not necessarily one cell, it could be 2 cells. So that's why we call it a colony-forming unit, as opposed to just one organism that causes a colony. I know it sounds kind of confusing. And let's remember, organisms have different requirements. No one medium is going to grow everything under the sun. So you are going to see a lot of different media in your lab. So same thing with time as well. So this is just an example, just to throw in here. If I can show you all stuff that I've done in the lab, I'm always going to bring that into class. Just so that you can see, what do people do who are actual biologists for this? What sort of experiments can you do? So I took a plate, and to save time and money, all sorts of things, I did 12 samples on one plate. So you can adjust these protocols too. It's not like you have to follow the directions all the time. You're in charge. You're the scientist here. So I did small samples in little bits of a plate, and I ended up with a plate like this. So again, this is what I would have done on 12 plates, but I had to do this for like 20 or 30 at once. And you'll notice that these are little black colonies. So colony morphology, color, shape, all depends on different organisms. All right, I think this is the last slide here. We can also count cells using a spectrophotometer. Big word. That just means we're going to put bacteria in a see-through container, okay? And we're going to we're going to shine a light through that sample. If there is nothing present, then just like in A, all of the light is going to pass through it. If there's stuff present, bacteria, yeast, fungi, microorganisms, they will absorb the light that would go through. So less light goes to our detector. So that tells us we have more cells present. So less light passing through means more cells are absorbing the light that you shine. So higher optical density, we're going to measure this in terms of optical density, OD. It is a function of cell number. More cells, higher optical density, less light passes through the sample. And this also measures live and dead cells. So what phase of the cell cycle would you want to take this at, or the cell cycle of the growth curve, would you want to get viable bacteria here? Yep, so if you measured cells, cell density during the log phase, you would get mostly all live cells. So you would get a very adequate estimation of your viable cell count if you took it during the log phase. That would be the type of question that you all should expect on the exam. So instead of just straightforward stuff, like I said, you're going to have more critical thinking. So which part during the growth phase would you expect the most amount of dead cells through a spectrophotometer? So I'm going to take this concept and put them together. So during the death phase or the decline phase, you would get the highest amount of detected dead cells. Does that make sense as far as the types of questions that I'm going to ask? Cool. So tomorrow we're doing chapter 6, and then we're going to skip the genetics chapters, and we're going to move into chapters 9 and 10 on Wednesday, Thursday. You got your quiz. 2 on Thursday. Between now and then, let's download the Respondus lockdown browser, and let me know if we have issues with that over the next couple days. If you all feel like you're struggling, you just don't know what you're doing, I need you to reach out to me. This week is the time to reach out. Our exam is going to be next week. I feel like that's going to be too late to play catch up. So please, please reach out to me if you need help. We can meet through HD meetings. We can do one-on-one meetings, whatever you need. I need you to tell me though. So thank you all for your attendance and your participation. See you tomorrow. Yeah. Let's just go ahead and say that because this is the group of people that are coming in. Yeah, y'all are good. Because on Tuesday, I'm only having 2 people. Yeah. So people online, do y'all have any questions? Anything at all?
Don't think so.
All right, cool. Well, I'm going to go ahead and end this recording. Please reach out by email if you have any questions. Like I said, we can meet one-on-one through HD meeting. Anytime you all need, please reach out if you need help. Okay? Thank you.
I have a question real quick.
So I have a question.
Yeah, well, yeah.
So I was looking at the syllabus to like do my readings for today.
Yep.
And for week 2, with today's date, it said pages 282 to 315. So that was chapter 9. So were there changes
gotcha.
to the syllabus?
Yeah, so I changed the syllabus and reposted it on the first day of class.
Oh, okay. So I just haven't.
Okay, so double check that. It is updated though on the home page, and the PDF is updated as well. You'll also notice that the quizzes only contain 10 questions now. I've changed that in the syllabus too.
Okay. All right, cool. Thank you so much.
Yeah, thank you for paying attention. I appreciate you. Was there somebody else?
Yeah, I have a question.
Okay.
So for the generation time, we will have for the quiz. So we will have like word problem to answer it.
I'm sorry. Can you repeat that question, please?
So for the time generation, or generation, yeah. So we will have like word problem to answer it on the quiz, or no? Because in the end of the chapter, it says like we have like a word problem, and we just calculate and get the number.
Okay, I'm having trouble understanding your question. So we're talking about doubling time, so generation time.
Mm-hmm.
And I'm sorry, can you just please repeat your question one more time?
So we will have like on the quiz. So we will like calculate like the doubling, like we will have like word problem, or no?
Oh, okay, word problem. Yeah, you might have a word problem. So that would be more of an exam type question where I could say, let's say that an organism divides every 30 minutes. How many daughter cells would be present after 4 hours?
Oh,
so that would be a word type problem. Or for the quiz, you should expect more of a straightforward definition type question, like what do we call the amount of time that it takes for an organism to go through its entire cell cycle? And that would be the generation time or the doubling time.
Oh, okay. All right. Thank you.
Yeah, you're welcome. So I'm going to make questions a little bit more straightforward, definition-based for the quizzes, and then a little bit more difficult and critical thinking-based for the exam. So yeah, you might expect a word problem for the exam.
Oh, okay. Thanks.
You're welcome. Was that all?
Yes, thanks.
All right, great. All right, anybody else have questions? Okay, I'm gonna go ahead and end this recording. Please reach out if you need me. Thanks, you all.